Welcome to Taxonomy Tuesday, everybody. My name is Chris. I'll be your host this week. We get together here, show and tell style, to try to help each other out and figure out what we have found during the week or growing in our lawns, Jim. And would anybody like to share from their device to get us started?
Chris, did you get my email?
I certainly did.
Okay,
Would you like to start?
sure.
Let's do it.
Okay, it's a weird one. um, I found these growing in my backyard, very sandy soil with a lot of gravel, like gravel in it. And this is what they look like. They look like they were growing on a root, but there are actually no trees in this particular area. They were old trees many years ago, but I think the roots that are in there are just decaying. It's a small area isolated from the big trees. But this is it took a very long time to grow. This thing was came up actually even a little bit before that heavy rain. By the way, we had 3.5 inches of rain here last Sunday, between the storm and the aftermath. But they were still growing through all of that rain. It took them a long time to get to this stage, and these pictures were taken today, this morning. They're finally up high enough where they're open, and you can see the gills. You can basically see the stem. Um, but the thing is that the most striking thing to me about this mushroom was the odor. It smells very, very floral, almost like, I think I described it as I wouldn't say cinnamon, that's a bad word. Carnations. You ever smell carnations? Carnations with some spice added to them. Very strong, like I could smell it from a distance. I mean, a foot or two away without having to stick my nose into it. I've tried to get as many different shots as I could here so you could see there's a cross section. And
Yeah, that's good stuff.
yeah, the original, or the younger ones, the gills were white. And as they started to age, they started turning into a tan color. And the spore print, I can't vouch for the exact accuracy of the color, but it is brown, but it's not a rusty brown like a Cortinarius would produce. So it's more like what an agaricus would produce, which is sort of a more chocolatey brown. But the mushroom itself is fairly tough. It took me a little effort to make a section of it. But there's the young one with a slightly older one just behind it. And you can see these are the ones I made the spore print of. And just strange that number one, they were very slow growing. Number two, extremely fragrant. And I've never seen them before, at least not in my yard. And I don't ever remember seeing them on any forays either. You'll note that the gills they're not crowded, but they're not far apart either. It looks like they are not attached, but I could be wrong there. If you take a look at the cross section, you can tell me, does that appear to be attached or not? No, does not appear to be attached. Anybody have any ideas?
Did you know what kind of root might have been underneath there?
There could have been old oak roots. There could have been old sassafras roots. I'm in the Pine Barrens. We're on the edge of the Pine Barrens, so our soil is very much sandy and in keeping with, a lot of the decaying organic matter is usually oak leaves on my property. I don't have too many pines on my property. I've got a lot of red and white oak on my property. So,
How about Hebeloma?
I was
hmm,
just thinking about the same thing. But the mushroom here on the right
Yeah.
is white.
mm,
So it kind of matures, to brown, gills. But when it's young, it seems to be whitish here, right?
yes.
And
What?
Hibberellomas smell just nasty. Either they're like radish or they have some other odors,
You know,
but
this
not
is this
floral.
is beautiful. I mean, it smells like flowers. It is really amazing.
But didn't you say flowers with a little bit of
Little spice, you know, like maybe
spice?
like a little allspice on it, or a little nutmeg on it, or something that would give it a little more of a slightly spicy smell. Not so much like not like cinnamon. I mean, for example, I mean some of the tricholomas, some of those have a cinnamony smell. This is not a tricholoma smell. It's a much it's a very strong smell. I'll say that, but it's not unpleasant at all.
Did you say the spore print was brown, or you obtained no spore print?
It's brown. Yeah, that spore print that we had. If you want to jump back to that, Chris.
Yeah, so
Yeah,
it cannot be a tricholoma then.
yeah, it can't be. I knew it wouldn't be. You know, once I saw that spore print, I was like, "Oops, tricholoma's ruled out right away."
Well, I would go with Brandon. I think it's some kind of Hibberelloma,
Eviloma. Okay, I'll
possibly.
do a little research on that and see if maybe I can narrow it down.
Yeah, well, Henry Becker has a beautiful Hibberelloma website in the aftermath of all the whole genome sequencing of all the type collections
Hmm.
worldwide. So that's like the utmost authority on Hibberellum we can get. There's nothing beyond that.
Okay.
And I think there's probably 12 species of Hibberellomas in New Jersey that we know of.
Okay.
So it's hibberelloma.org, and you can make some inputs in there, at least get some information on what we are actually finding in New Jersey.
Okay. Appreciate the
And then you
advice.
click on individual species and see if they fit your morphology.
Yeah. Yeah. You see, the cap has some scales on it. It seems to like to rise up out of the dirt with the dirt staying on the top of the mushroom. So, but I say it's a little tough. I mean, it definitely has a rubbery kind of characteristic to it.
Could we
Okay.
please go over the entire collage of the photographs one more time?
Yeah.
Yeah, I think the note about the gill attachment, which is it free? Is it notched? Um, that's often something I come back to with Hebeloma,
Hmm.
in my opinion.
Okay. Yeah, it
I do
appears
think
to be free, but it could be notched in just a little tiny bit of attachment.
right, yeah, exactly, maybe, but they also could be free. So
Mhm. I'm curious to know what the white is underneath the gills there. Just the coloration of the stipe, or it's not spores because the spores are dark.
It's just the white apex. It's
Yeah.
common in
Okay.
mushrooms to have an apex color differently from the rest of the
Right.
stipe. You see that in boletes a lot.
Okay.
yeah, just kind of like expanded right at the
Can
end.
can we zoom in, please, here?
Which one you want?
Or this photograph specifically. Yeah.
Now,
See how scaly the caps are?
Yeah, they're very scaly.
How big were these?
They were about the ones you're looking at here are about 2.5, 3 inches across.
We found something in the New Jersey Pine Barrens. So what for it was? The one in the south, which is always at the end of the season we
Alpine?
go to. No, the one near Mays Landing.
Okay. Oh, I know what you're talking about. Yeah.
We
Hill
we
Manor.
found a thing in there, and we got it sequenced, and turned out to be some kind of weird Inocybe, and the caps looked like exactly like that, like Inocybe
Oh,
species 19.
oh, cool. Yeah, that's not a bad idea.
So if you go to Mushroom Observer and look for that name, Inocybe SP Indiana 19, I think, you would probably see something that looks very similar to this.
Mm-hmm.
Was that from Wells Mills, maybe?
No, I didn't say Wells Mills. I
Estelle
said the
Manor.
Maze Landing.
Yeah, Estelle Manor.
Estelle Manor, yeah.
Cool.
So let me actually try to find the species, and I'll post the link, and you'll take a look. How about that?
Okay.
Just hold on a second. I think it's 19. I was just going over some of the results from that sequencing effort. No, it's a different Inocybe. Have to go into the species lists. I'll find it there, and that's going to be probably here. Inocybe species 61. Here it is. Okay.
Nice. And while we're still on this photo, do we all agree this is probably not an Agrosabe? Or is that kind of not out of the question also?
It doesn't seem to be growing in the kind of environment that I'd find an Agrosabe.
Yeah, and I'm not making the claim for it, just trying to,
Yeah.
kind of flush it out, make sure we're not overlooking, well, chocolate brown spores. That is kind of an Inocybe thing, or a Agrosabe thing.
Mm-hmm. Aren't most of the Agrosabe species tend to be spring mushrooms as well? Or do we have them all year? Do we have them all season?
Honestly, I don't know enough about Agrosabe. I think
Okay.
Igor probably knows more than I do about those, but just trying to make sure we're not overlooking something.
It didn't strike me as an Agrosabe, but I'm not an Agrosabe.
It doesn't to
Especially-
me. Just the form, stature and the form doesn't say Agrosabe to me.
Well, I put two links. I put a link to my observation and then to all of the observations of Inocybe species
Where do I
61.
find those? Where do I find?
It's in the
those?
chat. It's in the chat.
Oh, okay. I've got to call up the chat here.
Because the caps kind of remind me of that species.
Okay. Okay. Let me.
Since I'm not a member, I can't open that thing.
Let me take a look.
Which one?
Either one.
Oh, it wants me to log in as a mushroom observer member.
No. It's in it. Yeah. Well, if you're not
I'm
logged
not.
in, then
I
it's,
still haven't been successful and haven't tried really hard to get in. I had some troubles way back and I just never, kind of got frustrated with it and just never got back to it. I really should be in there. Okay, I can't get there. Okay, oh well.
Well, it's a shame that people don't use MO. Oh, that's all I can say.
Yeah. Oh, I have so many pictures and that sort of thing. I mean, I really should be using it.
No, I'm
Yeah.
just saying in general, like, not you specifically. Everyone basically left MO and went to INAT.
Uh-huh.
That's what people did.
Even not like Chris, you keep saying I'm not a member. It's like you just make a login.
I intend to, because now with all of you guys being able to just send all of your stuff there, but it's just something I haven't gotten around to.
No.
But that was
Yeah, that was cool.
that's cool. You got some good leads there, and
Well, as I get back into my mushrooming thing with my vision being better than it was, I think I may reattempt to get in there. So,
nice. Thank you.
okay. Well, thank you very much. I appreciate the effort. I will check out the Entoloma. I will check out anything else that I'll see if I can get into MO and check out these observations from Igor.
Yeah, you should, because I just checked them out. I absolutely see what you're saying there, Igor. Not looking dissimilar.
Okay, I'm just gonna,
Okay, so you're able to visualize these.
yes, I'm just
Yeah.
I'm just copying these URLs so that I can get in there later.
Would
And
anybody
Matt,
like to share from their machine, or we have other people who sent stuff into the emails?
Well, this forces me to join MO, and I really should. So
I mean, I can share from my screen, but if people want to see, because we have other things to look at, obviously.
yeah,
So
okay.
it depends on what people want to do.
If you want to show, Igor, go for it.
Okay, so let me try to share this tab.
There you go.
People see this?
Yeah. Wow.
So we have 3 observations, two from New Jersey. So this is Nina's here, the one which is 483, 647 on the right. And that's mine here. So if I just go into this observation,
Nina's looks so much like mine.
quickly flip, unfortunately I didn't take these pictures, at the foray. Took them on my rug at home.
Oh, so
So
they
that,
had a chance to decay some.
well, they just darkened, but they're already dark, they were maybe frostbitten or something like that. I don't know, even though this is from October. And this one is from Florida.
Yeah, look how white the one on the right is there.
Does this fit the profile of your mushroom, Jim? Does this look similar?
It looks. This one looks similar, but um, the older gills are not quite tan to me here. They're
Oh,
they're sort of an off white.
let's try then another observation.
Yeah.
Oh, this is Nina's,
Nina's
okay?
observation, they look tan.
Yeah, that looks pretty
That
similar.
looks very close.
Can we actually zoom in on the gill attachment on the one on the upper, the top one there, if we could?
Let's see. That's as much as I can go. They look to be attached. Is that the one they're looking for here?
Yeah,
The one
yeah,
at the top, correct?
correct. Yep.
Yeah. Yeah, this is the fully expanded view, and actually it comes as a separate web page.
Yeah.
I cannot increase the magnitude beyond that, but you see
If
the
you go
scales on the cap?
down to show original image,
Well, this is the original image.
oh,
Nina,
you already clicked on
Nina
that.
doesn't always
Yeah,
show
I already
that.
clicked and expanded it to the max. Like again, it's a separate page. Okay,
Hmm.
you're viewing the original JPEG,
All right.
How
which
about
is
that?
968 by 1200, pixels, whatever they are.
Yeah, that's not very big. So,
Well, I mean that's
mhm,
Nina's photographs, from the iPad, so she
right.
doesn't have a super duper advanced camera. So here's another photograph, probably in a fresher state.
Look at where the cap meets the stipe right there.
See that white?
Hmm. Yep.
Here we go again. Yeah, the gills are attached. They are actually adnate,
Hmm.
and you got the whitish apex, and then the brownish stipe, and
Hmm.
then the scales on the cap. So I mean, just an idea. I
Yeah,
think this
that's
chances are this is what we're looking at.
yeah,
Was
it
there
looks
any
pretty.
mention in any of the observations about the odor?
Let's see what Nina says about this. Usually she's good about the notes. Smell like red hots, the candy.
Ha!
Cinnamon,
Ah,
spicy.
spicy. I didn't see any red staining on the stipe.
No, there's no red staining in step. I said it smells like red hots,
Yeah, which is a candy.
but
Uh,
it's an odor. It's not the color.
yeah, smells like red hots.
It says further there, some red staining on stipe where broken.
Yeah, some red staining on the step were broken. Mixed woods, side of park. Yeah.
Interesting, when it says side of Park Road, that's kind of like what this soil is like in the area where I found these. It's almost like that sandy, gravelly type of soil with this decayed oak leaf
Yeah, so
mixed into it.
this is from Belle Plain, which is in the Pine Barrens.
Okay.
That one is from Bass River State Forest. I was wrong about
Mm,
the other location. It's actually basically just north of Belle Plain State Forest, different year, 23. And the Florida species, of course, that's a coastal plain.
okay.
So
Do you
it
need me to
appears to
save
be,
these? Do you need me to save these for any reason?
well, I mean, you could dry them. I mean, we could try and sequence them, but I don't know if it's going to be worthwhile. But
Okay. If
if
you
you could
don't think
dry a
it's gonna be
specimen,
worthwhile, I don't want to. I don't want to. I'm drying other things in my dryer right now, so of course these mushrooms will probably still be here for a few more days because, like I said, they're very slow to grow. So,
well, I'm going to stop sharing so we can proceed
okay.
with.
Thanks again, folks.
Thanks for sharing.
That's a good one good one to start.
Does anybody else have anything from their machine? Otherwise, we have Hunter's got some stuff. If we go in order of email receipt.
We could. We could do that, or I can wait too.
Let's do it.
Okay. So, my finds are from Wisconsin. I was there over the weekend. It's very dry where I live in Illinois. But I went up, and I shared these on my phone. That's all I've had time to do, and no research. So this is a cool blue polypore. And I kind of took it with these other mushrooms to bring the blue out, but it was growing just to the left of the other mushrooms. But I guess I only want to talk about the blue one, but there are, of course, lots of mushrooms here.
I know the blue one.
Oh, good. Okay. Well, so is it a hydenellum or is
No, Albatrellus.
okay? That's what I was hoping. That's what I
Albatrellus caeruleoporus.
Wonderful. Okay, good. It was one or the other, and I've seen that albatross before, and it was like I wasn't sure. So,
They grow in Stokes by Kittle Field, by the way. A
oh,
large
really?
thing.
I didn't see them.
Yeah.
Did I meet you there?
No, not this year.
Oh, okay.
Been a few years since I've been up there, so 'cause
Oh, okay.
I
I
live
just
in the
wanted
Pine
to make
Barrens
sure.
now.
Okay, cool. So that is the albatross. Awesome. Thank you so much.
No problem.
And yeah, I love seeing blue mushrooms. It's
Not
pretty
many of
nice.
them out there.
So I did take that. So I have that. It's like
We
loose,
have one at the top.
loose in the car somewhere. But I should be able to retrieve it, but I don't know if I'll do anything with it, but it's pretty cool.
Mhm.
So, and then I think that's probably since the other mushrooms are there, some tremella maybe, do you think? On the orange, and then some foliota. What do you guys think?
The Foliota kind of looks like Desarmillaria.
Quite likely. That may be it. Okay. Yeah, and hard to tell with a yellow probably.
Yeah, and
A little bit dried here.
I can't tell if the thing that you were calling tremella is that even growing on wood or is that just right on the moss there? Because that might be a
What?
slime mold.
What's
Uh,
this?
that's wood. It's a tree.
So we're right at the ground.
Yeah, at the root, like right.
Yeah.
See how the clump of orange looks like it has like a snail trail.
This?
Yeah.
Yes. Thinking more slime mold. Huh?
Maybe it just doesn't look like it's growing on a stick. I think.
Well, true. Should be if it's tremella, right? Um, yeah, it's too bad the photo's not a little bit clearer. I apologize. I'm gonna turn my phone on and see if any of the others are. But at any rate, really the blue one was the main one.
I'm glad Jim was here because I wouldn't have known that one.
I am
I've
too.
never seen that, Jim. It doesn't grow in South Jersey, does it?
No, I've never seen it down here. I've seen it. The only place where I've really seen it is up in Stokes along the Flatbrook and along the tributaries of the Flatbrook. And it's actually an edible mushroom, believe it or not.
Really?
I've never had it, but according to David Fisher and Alan Bassett, it's an edible mushroom.
What about a dye mushroom? Do you know about if it is?
That I don't know.
Okay. Oh, how interesting. Thank you so much. So this is Leotia lubrica. And this is an oddball. It's in Michael Quo's oddball category. So it's I think it's still kind of that's still where it belongs. It's commonly called jelly babies, and it's fun to see. And there's not really gills. There's not really pores. There's sort of line like some striations or sort of linear look where there would be gills.
Pseudo gills.
Yeah, exactly. And they're odorous and saprobic, I guess. But I mean, so they are fungi, apparently, as opposed to a slime mold, but I don't think they fit in the jelly category for whatever reason.
They are very slippery. Try to pick them up. They just slip right out of your fingers.
I did. I know. Yes. Um,
Well, I mean, jelly fungi is a construct of us, right? We classify things as jelly fungi. You know what I
yeah.
mean? It's absolutely. I mean, it's not a myxomycete. It's these are related to something cool, aren't they?
Uh, I don't know. I'm just looking at Mushroom Expert. That's the only place I've ever looked into them. But I don't know. But they are cool. I know that.
Oh no, they're ascos. That they're ascomycetes.
Oh, okay. Oh,
Yep.
cool. Wish I brought these home then. Darn it. I'll have to look. It would be fun to look at them under the microscope.
Mm-hmm.
Or
Yeah, they're. I don't know if they're common out where you frequent, but that species is super common here in South Jersey, at least, in my opinion.
Cool. There was a lot. I mean, it seemed like there was, I had more than one sighting up in northern Wisconsin, but here I don't get to see them too much. I think Quo was saying they grow mostly with conifers, but I have found them in oak woodlands. Yeah, growing hardwoods or conifers. Yeah. Okay. Um,
Oh, what is this? Clavariadelphus?
Clavariadelphus, and I haven't looked into what kind it is. Maybe Americanus, but I think it's the one with the L. Is it like Lignata Clavariadelphus? Let me look up real quick. I'm not sure, but these were so abundant. They were everywhere. I think you had said the Xyleria was like that earlier in the season, and I saw it similarly like that this year. These were like that. They were just everywhere, and anywhere there were pine needles, they were just all over the place. And I think this is like this is definitely one of my favorite genuses. I just love, I think they're so interesting.
They're so weird. They're weird. You know what
Yeah.
I mean? They're not. Yeah. I love them too. They're cool.
Yeah, I think it's ligula, Clavariadelphus ligula, is the one I'm leaning towards. And it's pretty much just based on the shape. Truncatus, it has more of a flat top, and Americanus is like a really nice club shape. And then these, I think, tend to be a little more lanky, and but otherwise really similar to Americanus. But yeah, so I just wanted to share some finds.
What
So
was the size of your club? Because the color is very small.
what was the size of these?
The clubs, yes, yes, because
Oh.
it only goes up to 3 centimeters for the color.
Probably 3 inches tall.
So it's too big.
For what? Ligula?
Yes, because that one goes up to 3 centimeters high, which is only 1.5 inches. Not even.
Yeah, well, I don't know. They were all different sizes, so some were bigger, but yeah, I do see like uh it says up to 12 centimeters tall in Wikipedia.
I'm just looking at Quo.
Okay, well, I'm looking at Wikipedia, and it says and up to 12 centimeters tall by 2 centimeters. So I don't know, but I haven't done any research at all. So you've done as much as I have today.
Well, I didn't look at Linicola. I've only seen ones that look like the Americanus and the one that they had named species before that one. Which
Oh
was,
wait, so this is ligula.
Oh, ligula. That's not even on it. Okay.
Yeah, this is what I think that is. Right. So he might not have it.
Yeah. They.
Yeah.
I'm looking at Myco Quebec. They have 2 to 10 centimeters. So.
That's like 4 inches at 10 centimeters.
Exactly. So these are well within the range for ligula.
Yeah.
And I mean, this is just Wikipedia, but they're saying they're widespread and common in coniferous forests, which is what was in.
Yeah,
but
that's better. Because
um,
the Americanus I see is always associated with beach.
yeah, and I mean, I think for Michael Quo, who lives to the south of me in central Illinois, we don't have much to speak of conifers except way down in southern Illinois, but
Huh.
much more deciduous. So I don't know. Maybe I don't know why he doesn't have it, but they're so interesting, so cool.
You just gotta send him the stuff and then he'll do it for you.
Maybe I think he sent out an email saying like he's not even doing that right now. I'll have to look back, but yeah, the last email, I'm on a list for like new names and stuff, and it was pretty it sound, I'll look it up. I don't want to say anything, but it did sound like he was like putting it on the back burner for a little bit. But I'll double check. I'll confirm next week or during the program later. So yeah, I love seeing these, and more the merrier. So this is a Suillus, and I is it I think it's either, I'm gonna get it wrong, but I want to say, so it has a dry cap. Um, it doesn't have a ring that persists at any rate. I think and it sort of grows in, like you see, kind of like the sphagnum. Um, I think I want to say it is Suillus clintonianus. Um, but I might have that wrong because again, I like literally just got back and kind of threw those up when I was out there. But yeah, I'm gonna. I don't know. Is Igor here? What does he say? Because these dry caps say no, I think. I guess he's not here. So,
Yeah.
okay. But Clintonianus is a larch Suillus. It grows with larch, and I think I found it as well, but the cap is more slimy on that one. So, um, Gravellii, perhaps, unless that's another name. So yeah, I don't know. I'm just showing off my pictures, which is terrible.
The
I
cap, the cap is not feathered, right? Like the spraguei.
no,
Yeah.
no. It does look textured though, or was it
Or
um
yeah, or hairy, hirsute, kind of
yeah, um
tufts of hairs.
yeah, or is it Brimos or
Not rimos,
no?
tomentos.
Maybe, yeah. Um, oh, that could be. Dry for sure. Uh, I will say.
Yeah, and I think that I don't think this is Clintonianus.
Yeah.
Suillus and boletes are not my thing. But I think that one uniformly is, has that weird cuticle and isn't going to have much texture other than just kind of viscid and gelatinous, right?
Hello, folks. I'm sorry, I had to take a phone call, so I was off, for a while, and I'm
Oh,
sorry to cut in, but I think
oh,
you were
thank
calling for
God!
me, right?
Thank God, because I was trying to figure out a Suillus that I don't know.
Yeah.
Okay, so this one is a large associate. I'm pretty sure of that, and it's not Clintonianus. I think it's ampliporus.
Yes, that's it. That is it. Because I found it last year, and I'm just going into my photos now too, and I could not remember the name. Just Clintonianus kept coming to me. That is it.
So let's see, Suillus ampliporus.
Nice.
I'm gonna type it into the chat.
Yay.
I can say I've never even heard that species name, so glad you
Well,
came back.
because we don't have larch here, it's a boreal species. It's all up, north, upstate New York and Maine and
Ah,
places like that.
yep, yep.
I mean, we're hardly. I mean, I found one Clintonianus in a park here in the middle of New Jersey that had planted larch that was dying. That's the only time I saw Clintonianus in New Jersey. But ampliporus, no, never found this.
Yeah, and yeah, it's not down here in Illinois either, but I did find it another year, and Arlene Bissett, I figured it out that it was ampliporus, and she confirmed, but then I completely have forgotten. But I do have a file in my computer somewhere, so I guess I'm not like afraid to be wrong all the time, like all the time. So I just rather show some pictures than hold back. But thank you. I knew someone here would know. So it's really cool, and it looks very similar to Clintonianus, except Clintonianus is like really slimy and has a ring, which is neat for a Suillus. Isn't that correct? Kind of like a slippery jack, the Clintonianus. Okay, anyways, we can move on.
And mushroom.
Okay, so this was a bolete I found in Hemlock. And I inquired from someone who really knows their boletes as to what it was, because I was hoping it might be something good. It was a lot darker in real life than the picture. The pores were quite dark brown, but and the cap color as well was really dark. But I believe this is Albarellus porphyrosporus, and I'm sure I'm saying that wrong. Porphyrellus.
Porphyrellus, porphyrosporus.
Oh, there's not a second P in that, or there is.
I'm typing it in.
Okay.
I think something like this.
Okay, yeah. So that's PHY, like a F sound. Oh, okay. Porphyrellus porphyrosporus. So, and
Are there any more pictures of this thing? Do we have any staining photos? Does this thing stain like other Porphyrellus?
I caught it and I do have more pictures, but because I didn't make it home before in time to send any, I just could send individual pictures from my phone, and I didn't want to hog it by sending a lot more than 5 attachments. So I should have included that one, but it did not stain at all when I cut through the center. And I might be able to just copy that into the chat, possibly. Do you think it could be something else?
No, I don't know this thing at all. I know Porphyrellus sordidus,
Okay. Yeah. And that
which,
one stains quite a bit, doesn't it?
well, it has an interesting staining pattern. It goes blue,
Red, right?
yeah, and then finishes red.
Yeah.
Or
That's
vice versa. I forget.
yeah.
But does this one doesn't do that? I guess, is that right, Igor?
I think it just may stain blue when you cut it.
No, it didn't stain at all when I cut it. I have the picture right here. I can try to copy this and show you. And I brought this one home as well. And so I can go look, and I was not really I was sort of hoping to go later. But here I am. So yeah. Yeah. Let's see. I'll show you the cut. I think we should come back to it. I'm sorry. It's didn't show up as quick as I thought. It's not here yet.
Where'd you? Oh, you
Oh,
didn't send it on your end.
I sent it to Igor. He might have a better chance
Oh,
of getting it up there sooner.
Well, I think only send me one or two pictures that, I mean, I can check my email right now.
No, I'm looking at your email that I sent you. That's why I'm kind of struggling because it's not from my photos, it's from this email attachment. But maybe on my phone it will work better, but I don't want to take the time. I feel bad.
Yeah, there is a picture with a cut, and it doesn't stain blue, but it should. At least in some, I think it's a species group, possibly based on some DNA inferences that were made by other people. They even given some provisional or cryptic codes to some of the porphyrosporus from the west coast. But the dark pore surface, I mean, it's overall a very dark mushroom, and
Very, very dark.
yeah,
Yeah.
I think it is a Porphyrellus, and it was pretty large too, right? It wasn't a small mushroom.
It was probably, I would say, like maybe 5 or 6 inches tall. So it wasn't huge, but yeah.
But Sordidus definitely stains, blue and red in the flesh and also in the pores. So we can definitely eliminate that from consideration. Uh,
Oh,
yeah, this is not. I would not call this one Sordidus.
oh, right.
Yeah,
Yeah.
it's not Porphyrellus Sordidus. You're welcome to take a look at observations of Porphyrellus on MO and INAT and see if any of them resemble this mushroom. Again, was this like in Wisconsin or
Yeah. This was in a hemlock forest in northern Wisconsin,
there
and
you go.
yep, and I'm putting the cut picture in the chat right now, maybe, hopefully. Did it show up? So,
Yeah,
okay. Is it open?
it's not blue.
Yeah, that's weird. It looks weird. I'm gonna try that again.
I can share the photo maybe.
Oh, here, this time it worked. I believe. There, it should come through, I think. There it is.
Oh, wow. You put the picture right in there.
Yeah. Yeah. I don't know what that other thing is, but
Oh,
Well, either that or it's some kind of Tilapilus, but I cannot think of a Tilapilus that looks like this that I know.
yeah, I mean
Uh,
the pores are much darker in real life.
the pores are really, really brown, and no Tilapilus is going to have pores like that when they're young.
Yeah, I'm wondering if this is the gills is maybe like some staining, unless they're actually pink, because
They do look a little pink there, don't they? Yeah.
Well, the tubes are going to look pink Porphyrellus is a sister genus to Tilapilus, and they sit
oh,
in Boletoideae together with Boletus and other genera. So they're all fairly closely related to each other.
gotcha. Okay, yeah, because
Xanthoconium, Tilapilus, Porphyrellus, and Boletus are all next to each other on the tree.
yeah, and like I said before, my only reference point for Porphyrellus is Sordidus, so I'm kind of biased and like, oh, it's gonna stain, right? Yeah, no, not so much. That's just it has kind of pinkish tubes.
But now that Hunter mentioned that this is northern Wisconsin under hemlock, I'm pretty sure this is Porphyrellus porphyrosporus. There's no question about it. We will never find this thing in New Jersey. It's too far south.
I'm gonna look at it now. It's loose in the car in the car somewhere. But I'm curious if what it's looking like now, and definitely what it's gonna be smelling like if I don't get it out of here.
That when you said that earlier, it's loose in my car somewhere. Man, that I relate to that so so much. There's just dried up mushrooms in my car in every corner.
Yeah, yeah, it's sad that they don't make it out of there sometimes, right? Yeah. All right. Yeah, and I don't think I can find my box. So I'm so sorry I'm not better prepared. What's next? What else do I have? We should move on. I got
That
one
that
more.
was all of them you had there,
Oh,
I think.
good, good. Hopefully so. Good. Okay. Well, oh no, I thought I had a Lactarius, no?
I don't think so.
Oh, bummer. Okay. Well, I got lots of good stuff I can show next time, and I will. Or if we run out of time, I can get another email together or something better. Oh yeah, you found it. Okay. Cool. Let's see what else people have.
Let's see who's up here. We got one from Doug. You want to go?
I think he's doing something, but he's there.
Oh,
Yeah, can you hear me now?
there he is.
Yeah, I had a little trouble getting my cell phone to work. It's getting kind of beat up and old. Uh, I was up in Maine about 10 days ago in the extreme northeast corner visiting some old friends. And one afternoon, we went out to a place called Quoddy Head State Park. It's a little peninsula that sticks out into Passamaquoddy Bay, south of the town of Lubec, which is due south of Eastport across the bay. You can look out over Grand Manan Island from Quoddy Head State Park. It's the foggiest area in the U.S. And while the rest of the state was extremely dry, there were some mushrooms right along the coast that was totally enveloped in fog when we were there. And I didn't see a great number, but I saw a couple of these. I wasn't sure. I thought maybe one of the experts like Igor could comment. Um, it had this white netted reticulation on the stem and this kind of ochre rusty brown cap. Something had taken a bite out of it. And let's see. I think there's two more somewhere. Do you have yeah, those other two photos, Chris? Yeah. This is the undersurface of the cap and one that had fallen off. Very dense pores, pure white, didn't seem to bruise. And the third one just shows from the underside. Uh, it might have had the littlest hint of yellow on the underside of the cap. It's a little bit an artifact of the shade. It was mostly whitish. Um, and that's what I saw. I kind of thought it's Boletus edulis complex. I saw that there's something called Chippewaensis up there, was underneath. These are pretty much all conifers. They have a couple different spruce species up there, and they have hemlock, quite a bit of moss and ferns in this area. And the ground was damp, but it had not rained in a while. So Boletus edulis complex is all I could come up with. I don't know. They have Varipes.
There is no Boletus edulis complex. It's only edulis. There's no Chippewaensis. You can throw out all these old names away. Dr. Dentinger from Utah, in his sweeping study, has established that it's a highly variable Holarctic species. And the
What's that
European
word there?
population of edulis actually is the youngest one. And the oldest one is from Alaska. So it traveled from west to east across the globe. And that's what it is. It's our northeastern edulis.
Yeah, I'm gonna piggyback off of Chris's question. What was that word? Hollow Arctic?
Whole Arctic.
Whole Arctic.
Yeah.
Whole globe.
Well, there are different regions, like temperate region and maybe Arctic region and subtropical. It refers to the climate belts
Gotcha. Gotcha. Okay.
in any given hemisphere. So
HAL, Whole Arctic, or OL?
whole Arctic, HOLARCTIC.
Hmm.
Thank you.
It's relating to or denoting a geographical region comprising the Nearctic and Palearctic regions combined. And you just go into Google and it's gonna show you a map. Basically, it's like pretty much the entire temperate northern hemisphere.
So that's it. Edulis is edulis, and that's it.
Edulis is edulis.
Yep.
Yeah,
Yeah.
So all those fancy names that they came up with, for Eastern and for Western, like edulis variety, grand edulis, it's the same thing.
Hmm.
What was that species? Rubriceps is also edulis.
Yeah, and Doug mentioned Chippewanensis, which is something I found up in Michigan that
Yeah,
I wanted to. I was dead set on calling it Chippewanensis, and nope, Edulis.
yeah. There's no such thing as Chippewanensis. It's been folded into edulis.
Is Atkinsonii still a name?
No, Atkinsonii is from a different branch of Porcini sensu stricto. That's an oak-associated species, and
Oh,
it sits in a different clade.
got it.
It's from the Arius clade, and Arius is a species from Europe. So things like Varipes, Atkinsonii. What's the other one? There's one more. They are all from that other clade, but it's still Porcini sensu stricto, but it's not edulis.
Okay, but like Boletus separans, for example, that's still Boletus. It's not
That's still
Edulis.
Boletus, but it's not Porcini sensu stricto.
Right.
It's a
Okay.
different,
Cool.
totally different clade. That's not a part of the edulis clade in a broad sense. Yeah, I mean you have to read the papers. It's quite complicated. Um,
What kind of genetic variation is there within Edulis as far as
well,
DNA sequencing?
I mean ITS is going to be the same. They sequence whole genomes, and they found out that there are admixtures in certain populations, but by and large, it's one single species that they just marched along the globe, and somewhere, it got maybe slightly segregated from the rest, and you got pockets of populations that have slightly changed over a long period of time, but genetically it's edulis. Like rubriceps from the west coast, it's edulis. There's no rubriceps anymore.
Hmm.
You have to go and read the paper by Bryn Dentinger, and if anybody interested, I can forward some links. But it's a rather thick paper, a lot of science in there.
Yeah, that would be good if you could forward the link to it. Thank you.
Yes, it would be nice to bring him in for a talk. He's pretty good with talks and stuff. It'd be good to see a new updated one.
Well, he actually gave in November of 2022. He gave an hour long YouTube presentation. I was there. I mean, it's not YouTube. It was a Zoom, but it was published or posted to YouTube,
Yeah,
and I can fish out that link and provide it here. So therein he discusses Edulis as well as all of the Porcini or Boletus species in North America, what the names are.
right. But the paper's even more up to date, right? I mean, wasn't there? He wasn't done then, right?
Well, it was essentially that paper was being worked on. I mean, the paper published in 23, whereas the talk was in November of 2022. So when he talked, he basically paraphrased his paper
Oh,
that
okay.
was at the time not published yet, but the draft was already written.
Yeah, that is a good talk. Yeah, it would be good to see that again and read the paper.
Yeah, so I'll put both of those links into the chat.
Great.
Cool. Thank you, Doug.
Doug, did you eat that?
Oh, no, no.
No.
Just hiking for the beauty with some other people who were not mushroom aficionados or foragers, and I just happened to see that on the side of the trail. Spent more time looking at the absolutely fabulous scenery there. You know, Maine is a beautiful. Most people don't go past Acadia, which is quite nice, but it's overrun with people. And you get out east of there, and the scenery is just as spectacular. And you could hike for 4 hours and see one or two people. And the big steep cliffs crashing down to the sea with bald eagles and seals. It's quite a beautiful area.
Sounds amazing. I know Lila was just out there a couple weeks ago, and she said it was very dry, but I don't know if it was dry where you were, but where she was.
Yes, it was dry all over. As I said, just this in the headlands that abut the seacoast, you tend to have fog. It sometimes will burn off in the afternoon, but it didn't this day. It was just completely enshrouded in fog, so it gives kind of a bit of a damp ground. But if you get in from the coast during this droughty time, and they've had very little rain the last 2 months, even 100 yards in, the moss is brown and dried up. So it's really just along the coast that I was able to see any fungi.
Yeah, in the drip line or whatever. That's good. I'm glad you got to go there. Sounds amazing.
Yeah, I hope you can visit it. It's a beautiful area.
I want to.
Cool. Martin has a good one. Were you still around, Martin?
Yes, sorry, I was on mute. It's probably not intellectually interesting to you guys, or just curious because it's in my front yard and it appears every year.
I'm on my I don't know. It's gotta be almost a dozen in my yard now.
Yeah, there's lots of puffballs as well, but this didn't know what it was. And I didn't do a spore print. I just put it on a plate. It's in the fridge. I put it on a plate. It seems to have very detached gills. It's just growing at the boundary of the driveway and the grass.
Oh yeah, those are no question free, right?
Right.
Got a good bit of separation there.
Chlorophyllum?
Yeah. Yeah, it is. It doesn't look like mollidites. The stipe seems a little slender for that. So maybe this is that other one. And I don't know these things all that well.
What
But I
kind of
like.
trees was it, Martin?
It was just in grass next to a driveway. I don't think there's been trees there for many, many years. I don't think there's any trees associated with it. I think it's like a field mushroom, but there is a underground stream under there. But I doubt that makes much difference.
Mm. And you're in New Jersey?
Yeah, this is like Tewksbury, New Jersey.
I know Lila and I found this macrolepiota that I was like Procera, but it was so short, the way this is, and it kind of reminds me of that. I mean, I think
Yeah, that's the other option, right? Macrolepiota. Um, but
Yeah, there is
I
a species besides Procera that is short, and she made an observation of it. I think it starts with an M,
yeah,
but
yep. Uh, I know the species you're talking about. But I think the Macrolepiota thing, don't they all have the annulus is like free moving, whereas this one looks like it's, pretty well normal annulus.
yeah, I know Procera's like that at any rate, but I'm not positive about the other one.
Yeah, I guess I'm not either. That's a good point.
Because I think if I remember correctly, the one she and I found, it also didn't move, but that was what she had gone with, name wise. This, instead of a chlorophyllum or something, but it was in uh the Great Swamp area. A
Yeah,
lot of litter.
yeah, and I think that is a Macrolepiota thing. Where Martin found this kind of, that's pretty prototypical for
Chlorophyll.
yeah, chlorophyllum, right?
Yeah, and molybdites in would, in grass.
Yeah, Rhacopus. Jim put it in the chat.
Oh,
Chlorophyllum Rhacopus is a good idea here, I think.
I thought that I found it before in woodlands, but I may be wrong. I would think there would be a tree associate with rhacodes. There's the Brunius as well, right?
Hmm.
But yeah, I don't know. The spore print's going to be very important. Also, molybdites. I don't. The scales can wash off, but was
Hmm.
this the only one, Martin?
No, there was two. They opened very quickly, like less than 24 hours, and it's about 7 inches across the cap and maybe 6 inches high or something like that. Definitely from just Googling, um, the chlorophyllum looks more like it just on the initial images than the macrolepiota to me.
Yeah.
Especially the ratio of the stipe width to the cap width looks much thinner on the macrolepiota than the chlorophyllum, but I don't really know that much about it, so I was just
Yeah, the macrolepiota has a really thin stipe. Yeah.
right. The chlorophyllum looks, the images I'm seeing also have the same patterning on the top of the cap as well.
Yeah, it's funny, like the ones that I've been getting because of the weather being what it's been, the dry, the way that it cracks around the edges seems to just be like that's what every single one of them eventually does by me, especially because it's dry on their way open, like these cracks here. And I don't know if other ones do that the same, but yeah, this one is spitting image of the one, except for the gills on this aren't, they don't have that marginate thing going on.
Yeah, I didn't do a spore print just because I naively assumed it would be white and therefore not interesting, but then I decided that was stupid, so I just left it on a black plate just now, but we'll see how it does.
Yeah, and the molybdites will have the green spore print, which is
Oh,
why it's be so telling.
okay.
Yeah, chlorophyll and molybdites, which is poisonous, and so the sickener.
Yeah.
I'll keep that in the fridge with all the food then. That sounds good.
But
Nice.
to that point, I have a thing that I'm going to share. I sent an email in. Um, assuming that it was going to be a boring white spore print, and it completely surprised me. So, getting a spore print, it's easy to do, and can tell you a great deal sometimes. Not all the time, but sometimes.
Okay. Well, thank you very much.
Thank you. Would you like to go, Brandon? I think you're in here.
Yeah, sure. Okay, yeah, I started with 3 Amanitas, um, and I kind of chose this name out of a process of elimination type thing, and it's not a great observation because it was getting dark and my phone was dying, so it actually, the next photo is the last photo before my phone died. Um, that cap wasn't as blue as it kind of appears. It was stark white. I don't know why my phone decided to make it look blue. Um, but yeah, this is the in situ photo. Conifers, pitch pines, pure pine barrens is where I found this thing.
What's going on here? Did it explode out of the ground?
So I think that's PV material. I think that's the partial veil. I could be
Correct.
wrong.
This
Okay.
is the partial veil falling off the gills, and this is what happens to any Amanita polypyramis.
Okay. Cool. Yeah, and like I said, it was getting dark, so I only got these 3 photos, and my phone died after that last photo. I had to charge it up, and then I took this one under a street lamp.
And the thing is, that's good enough for me to ID this as polypyramis. If you showed
Yeah,
this picture to me, I would have told you what it was.
cool. Yeah, yeah. Because I mean, it's so, you know, uh, I don't know how to even describe that. It's not rooting quite per se. It doesn't have any. Maybe it's all UV material. I'm not even sure, but it doesn't
It's
have
so
like.
it's so universal veil. All these pyramidal structures on the stipe, cap, and the bulb, which is not rooting. It's a tidy, almost like globular, bulb.
Mhm. Yeah,
Uh,
yeah,
uh,
yeah.
yeah,
Like
these are not
Oh,
radicating. It's
a garlic bulb. You know what I mean? Where
yeah,
it's just.
exactly. Like maybe slightly. Yeah, yeah. I mean, it's pretty much round. It's not even turnip shaped. Maybe
Mhm.
a little bit, but s- sort of subglobose bulb.
Yeah. Okay.
Maybe
Cool.
a little bit of a rootlet down there, possibly.
Yeah. No,
If it's
no. That's mycelium. That's
oh,
like a
that's my
myc.
medium. Okay,
Yeah.
okay,
If you
all
can
right.
zoom in a little bit, I think you can. Yeah, it's taken under a street lamp, but
Yeah, it's mycelium intermixed with the substrate.
correct. Right.
Yeah, yeah. But
Right.
look at those, subpyramidal warts,
Mhm.
tiny ones. This is very typical for the species. It just covers the entire stipe all the way down to the base.
And those subpyramidal warts, they're not. I described them as not structured, so like you could wipe them away with your finger. You know what I mean? Like something with like a nusta, for example, pyramidal warts. You couldn't wipe those away with your finger just by picking it up. So
No, but these are very delicate, I think, and soft. You can scrape them off, I think.
yeah, cool.
Yeah, this is exactly when this Amanita starts to show up. It's in the fall, and it goes all the way into I think October, maybe late October.
Can,
Nice.
hey, Brandon, can we go back for 1 second? Can you just open the range on that mushroom? Because I think it's just south and east, and I just wanted to double check it's not something over this way.
Yeah, she's talking to you there, Chris. Click on the map if you could.
Oh, sorry. Right.
And I also wanted to mention this thing is huge. I mean, uh,
oh,
I it's in the notes. I want to say it was like 14 inches wide or four. Yeah, no, 19 centimeters or something like that.
Yeah, Rod Talos on his website when I was working actually said that these can be the size of a dinner plate.
Mm-hmm.
How far out you want to go here?
Far out. I want to see
Go to
the,
your house?
yeah,
Yeah.
all the natural range, because I think it's just mainly south and eastern species. So I wonder about those northern finds up there, but okay, thanks. Yeah.
Man, how do I how do I get back?
Um,
Oh, wait a minute. Ooh. Aha.
the X.
Right.
All right, cool.
Thanks.
Another one that I feel better about this identification, especially because you have the purplish UV material and that kind of translucent looking cap. I struggle to ID Amanita lavendula without those features, and oftentimes they don't. They either bake out, they're not in perfect condition, so you can't see those. But I think this is lavendula.
Can you please repeat those features that you're looking for actual lavender color somewhere?
Yeah, on the universal veil, so the remnants of that on the cap there that he's highlighting, and then the translucent quality to the cap, um,
Cool. Thanks.
which which might not be the best descriptor, but that's the one that I keep in the back of my head.
Yeah.
And then the bulb as well is pretty unique for this thing.
What section is this?
This is Validate, right?
Validate, naturally. Yes.
Yeah.
A bulb, a ring, and no striations on the cap. And you can see that the ring kind of wore off to this gray, purplish gray patch on the stipe, oppressed. Yeah, below the needle.
Here.
Yep. So this is in the lavendula group.
Okay, so we can't even call it lavendula, lavendula.
No,
It's
we
the whole.
can't without more information like the DNA, an ITS sequence. We can't.
Okay,
Yeah,
but
I don't bother with
lavendula.
these. I mean, these are so common. Who cares, right?
Yeah. Well,
I mean, you
I
see
care.
these things
I
and you wonder if they're the same or if they're different. Yeah, but you cannot sequence every specimen.
Right. Yeah, yeah. I care about just adding lavendula group to the thing that I found this day, to the list that I found this day.
Yeah, but that's good enough. I mean, I mean, the thing is, it could be lavendula because it's one of the what 5 or 6 species in that tight cluster, and I think the Pine Barrens has a few of them.
Hmm.
Um, but it could be one of the more common ones. So chances are you could be 50% right, you could be 100% right.
Yeah. All right. I'll
And
throw.
as far as I
I'll throw.
know, INAD does not allow you to propose anything below an actual species, like a group or a complex.
You can go section. I could throw this into section validate,
No, but
or
that would not be useful. I think lavendula here is a much more useful name. Yeah.
all right, or lavendula group. Maybe they have, which would be maybe useful as well.
Yeah, recall that on the Mo, this is not a problem. They have a rank, in the names that is either like a group or a complex, and you can basically, call like Amerirubescens group, Lavendula group. You can select that name, and that would be fine.
Right. Yeah. Yeah. I don't think they have lavendula group on INAT, but they have a Amaryrubescens group on there, or a complex, something like that, that you can select. But can you just briefly mention some of the other species in the lavendula or lavendula group?
Well, there's
Just so I
this
can write them down.
Americitrina.
Americitrina.
Yeah,
And
and
that's why I was wondering, Brandon, what the smell of this one was. That's how I usually tell it.
Probably didn't have a distinct one. If I didn't put it in the notes, it didn't have a distinct odor.
Yeah, I didn't see in your notes. I didn't see any notes at all. There you go. Oh, no.
Yeah, I probably didn't put any in.
Okay. Thanks.
And if I didn't put any in, I didn't note any odor.
should smell like potatoes or something.
Yeah, well, the amanitas that are supposed to smell like potatoes have never smelled like potatoes. Like that Saliolins
Sauvignon, yes.
that I sent you that email about. I didn't get an odor off of that, and that's supposed to smell like potatoes too, right?
Yes, that's supposed to smell like potatoes, raw potatoes.
Yeah. All right. So moving
So
on.
you asked for names.
Yeah, if you could just
And
talk about them real quick, or just mention them real quick so that
yeah,
I can
so
look them up.
Americitrina, Lavendula, Lavendula, Corneli hybrida or hybrida. So Corneli like Cornelius, and
Yep.
hybrid hybrida. So that's also
Okay.
provision name. So 1, 2, 3, and also in that stirps we have Porphyria and Seleniolens, but those are not, in the strict Lavendula group.
Right.
Those three I can see right away. So then the European species would be Amanita mappa. That's MAPPA.
Series mappa. These things are going to be in, right?
Yeah, that would be in series MAPPA. Correct.
Okay. Cool.
And I can drop probably a link. Yeah, I can drop this link here to Rod's website I was able to find through Google, except that the website itself is dead. But when you start looking or searching for species, an individual page is gonna show up, and you can actually open it,
Yeah.
which is I think is weird.
Well,
But if you go to amanitaceae.org, it doesn't work. But if you click on this link, it's gonna work.
A specific thing about Amanitas, his page pops up and you can actually view it. I noticed that too recently.
Yeah, I think he might have moved his Amanitaceae to this other website called Tulis Labs or tulabs.com.
All right, cool. Thank you. Let's go on to this thing that I'm quite certain is section Roanokenses. But yeah, I am wrong often. So this thing, I didn't take a photo of the cap because it was just covered in sand and it was a confusing photo. But yeah, you can see it's got that taproot, the turnip-shaped rooting stipe to it. Really handsome thing. I got no odor off of this one either, and I'm all the things that I was looking at trying to put a species name to it. They all had specific odors, so I didn't apply a name to it. I don't know. Roanokenses. These things confuse the heck out of me. The gills here are decurrent. I think there's no other way to describe them as decurrent.
Yeah, they're subdecurrent. They're not free. I mean, this is highly unusual for the Amanita. I mean, sometimes they look like they're attached when young, but as the mushroom expands, you get the free gills. But this is clearly, looks like something like, like a hygrophorus or something,
Mm-hmm. Yeah, yeah. But it is, I mean, it's an Amanita, right? It's section Roanokenses, do you think?
That's what I thought, it's just like a core lepidella.
Yeah.
That's where most of the lepidellas wound up in section Roanokenses. Mycorrhizal lepidellas, that is.
Mm-hmm. Okay, that wasn't this one. This one's mycorrhizal, I'm pretty sure, based on that taproot. It wasn't attached to anything, and it was in sand. If you can go back to the last photo, you can see the same thing that we were talking about with, or maybe the one before, with my finger there with polyramis, how all that UV material is just kind of dusty. If you notice on my finger, it came off when I was just handling it, as well as this, kind of inconspicuous annular region. If you can zoom in there.
So the partial veil in this species was very evanescent and almost like a cortina-like sort of woolly flocos. I'm
Yeah,
not sure
yeah,
how to
yeah.
describe it the correct words, but it was almost like, yeah, like like a cobweb. And as the cap expanded, all of that sort of fell off, and it left a ring zone on the stipe. And one of the species that I think does that is Amanita longipes.
Longipes. Okay. Does that one have a distinct odor that you can remember?
No, but there's a similar species that Rod Tallis called chlorinosma, and that one smells like chlorine.
Right.
I've seen things like that in the Pine Barrens, in the past, and I actually have observations of them. But recently, I was able to sequence two of these things through, an effort with the Ohio DNA lab,
Mhm.
and one of those was allegedly longipes. I'm not sure what reference they used for that sequence, and the other one was Cineriaconia species Florida something. So to me, they kind of look pretty much the same. But what do we know about these lepidellas anyway? I mean, they're completely confusing, and nobody can identify them properly because we don't have type sequences.
All right. So best name for this thing right now really is just section Roanokenses, right?
Y- Yeah, I don't want to put any wrong names in this thing.
Yeah. Okay. Cool. Well, it's I dried everything from this day. So
Great. I mean, nanopore sequencing is the only way to go with these lepidellas, because Sanger is not going to give you an ITS with these things.
so
I
gotta send
guarantee
it
that.
gotta send it to the Ohio lab then,
You
right?
gotta have to send it to the Ohio lab or Steve Russell or maybe even
Yep.
John Plishke.
Yeah. Cool. Nice. Thank you. All right. I love this thing. This was so much fun. Because for 2 days I was looking at the wrong genus, trying to identify this thing while it was laid out for a spore print. Uh, yeah, everything's white except the gills are bright yellow. And then if you can zoom in on the gill attachment, anyone want to guess what genus I thought this thing for sure was?
Hygrophorus. Hygrossum.
Tricholoma?
Tricholoma. I thought for sure it was like a pale tricholoma, um, equestri type thing. Um, but and I got kind of giddy when it revealed itself, um, because it had a pink spore print. it's an Entoloma. It's Entoloma luridum. And even these Entoloma spores are unique for the genus. You can see the one that's lined up right on the if you zoom back in on the one, on the 5 there, you get a good
Wow.
measurement out of that, but you can also see the angles on it, right? So for Entoloma luridum, the spores are subglobose entelomoid. So they're Entoloma spores that from most angles look like a subglobose spore with an apiculus. But yeah, you can see they have angles. They're Entoloma spores for sure.
Very cool.
I was looking at pictures of Entoloma spores that somebody was doing under an electron microscope. I bet these are a real swell candidate for that.
Probably.
Oh, there you go. That's a winner.
Yeah, that's another thing.
You got all four on that one.
No, 2.
You sure?
So they're 2 and 4. If you can find a 2,
This looks
you're
like
on it.
1, 2, 3, 4.
I'm not seeing that. I looked at it for a while and came to the conclusion that it was a 2 sterigmate basidia, but I could be wrong.
Either way, I love finding them.
Yeah, me too. If I find one that shows any stigmata, I'm taking a photo of it. This is a throwaway. I'm not sure what that is. I thought it was a cystidia at one point. Same with this photo. You probably have to zoom a little bit. Yeah, right there if you could.
Hmm.
Yeah. Yeah, the one on the left, you can see kind of a ghost of a cystidia, if you will.
There you go.
Yeah.
That one there looks.
It looks globose, right? It looks like a balloon.
Yeah, but like, it's got that. It's probably better under the microscope too. It's got that like, what do you like a that geodesic look.
Yeah, that's a cool descriptor for it. You know, um,
Like points connecting.
right. Yeah, angles at any point. There are flat sides to this thing.
Right. But enough flat sides to make it appear globose.
Mm-hmm. Yeah.
Yeah, that's cool.
And that fits right in with Entoloma luridum, which is the name that I put to it. Dried orange, the gills did. And you can see this is my quick triage of fungi that I found this day, and I labeled it as tricholoma because, man, that thing looks like a tricholoma. And bad photo, but this is the spore print on the bottom of the Entoloma luridum compared to that's Cleidocybe, right? Ampulocleidocybe clavipes on the top, which is a white spore print, and then you can very clearly see it's a pinkish brown spore print. It's not white, so it's not a tricholoma. But that was the giveaway. When I saw that, I was like, oh my god, it's not a tricholoma, it's an Entoloma.
That's a cool mushroom.
Yeah, I thought so too. I really I like that one.
Yeah, I saw this a couple of times, and I only found this in the Pine Barrens. I haven't seen it anywhere else.
Yeah, and this like I said, this was actually an Atlantic white cedar stand in the Pine Barrens. You can see those kind of cedar needles or whatever you call those in the background there. Which the whole time I'm like, that's a weird place for a trichloma, right?
Yeah, you know what? My collection from Brandon Burns State Forest was also on the edge of a Atlantic white cedar bog that goes on that road off Cooper's Road. That if you go away from the Packing Pond, taking Cooper's Road sort of north,
Yep.
you in half a mile it'd be like across this near this intersection, and you just make a right, and it will go straight through a cedar bog, and that's
Right.
where I found this.
Cool.
So yeah, it's the same kind of habitat. Interesting. Two different places.
Yeah, and it doesn't seem to be collected all that often. I suspect because of where it's growing or the misidentification. Igor, I don't know if you remember when you found that thing, but I put it in my basket and I was like, oh, that's a weird looking tricholoma with a white cap and a white stem, but yellow gills. Must be an equestrian type deal, um, especially with the gill attachment. So cool.
Yeah,
Glad to know.
I'm looking at MO observations. So only 45 of them on MO of the species, provided that the identifications are correct. The most recent one actually came from Bass River State Forest, and you were there.
Hmm.
That was back in 2023, October 8th.
Oh wow, nice, cool.
And my collection from Brandon T. Burns State Forest dates to September the 12th, 2017.
Yeah, I remember looking at that exact observation from 2017 on MO.
And
So
my notes say, I'm going to copy this link and put it in the chat. My notes here say, growing singly or in doublets on the sandy bank of a gravel road cutting through a cedar bog.
hmm, yeah, great.
And here it
And then
is.
uh, this last one, just for the sake of time, I'll try to breeze through. Um, Very small. If you can zoom in on the millimeters on the measure there, you know, the largest one was about 2 millimeters, um, maybe a little bit more, but certainly less than 3. And all the other ones were much smaller. And this, much like the Entoloma we just looked at, was a little bit of an imposter for me because I thought it was an asco. I thought it was, uh, something in the Orbiliomyces family. But no, this is a Dacromyces. You can see it's got a little bit of a foot, kind of up to a cup, sunken cup. But here we have the tuning fork basidia with one spore per sterigmata.
Um,
Oh, there you go.
Yep. And all the other thinner uh, tuning fork type things, that's actually, well, it's a mixture of mature basidiums, basidia, and generative hyphae, which the hyphae on the generative hyphae on this particular species kind of grows wildly. It's pretty erratic. And then I think there's maybe one more photo of a tuning fork. Yeah, there you go. I don't know why they call them tuning fork Basidia.
Yeah, seriously, that could, you know.
Yeah, good name for it. That's amazing microscopy on such a small specimen.
Thank you. Yeah, and it's another case of just an underobserved thing. Like Dacromyces minor, there's very few observations using that name. But I mean, the thing I had to rehydrate. I had to rehydrate it to get it to 2 millimeters. You know what I mean? Um, so when I found them when they were xeric, when they were kind of dried up, uh, I thought they were Biliomyces, but they rehydrated pretty big, and you know who's doing that?
And what'd you do? You throw this whole stick in the thing, or did you pop it off and put it in
Uh,
something?
what do you mean to rehydrate it?
Yeah.
Um, so I sat that stick, that twig. I mean, that twig is less than a centimeter wide. You know.
Stick, twig, whatever.
Well, yeah. Well, no. I think there is a differentiation between twig, stick, branch, and log. There,
For
those are four
sure.
different things. But this was on a twig, and I just laid that in a layer of water in a cup. You know, I put maybe a millimeter of water and laid that twig in it. Left that for maybe 15 minutes, and then it rehydrated itself. So,
I've been messing around with the like little moist chamber culture thing, like uh
Dorothy, or was it Dorothy?
like Virginia's slime mold method.
Virginia. Yeah, yeah,
Yeah.
yeah, yeah. So man, she's onto something with that. Uh, and there's like a bunch of different ways I've been messing around with that, but I think that's super useful. And like, like instead of putting something in water, even just having it in that intense humidity, which can easily go awry. But
Well, that's good for slime molds because you need them to reach their next stage of development, right? Um, with when I'm rehydrating things like this, they already reached their fully developed stage. And the cells have dehydrated, so you just need to rehydrate those cells. Um, you don't need to coax this thing into the next stage of development, if that makes
no,
any
no, no,
sense.
I know, but like even as it's rehydrating, to like depending on what the thing is, sometimes it just really helps that it's not just wet, that it's also sitting in humidity, you know? Like
Yep.
I'll take like a stick like this and put it in and it's still touching the water and doing that, but I'll like throw a cloth over it for a little while and it seems to help.
Yeah, a wax paper bag is a great way to do that. You know, I have a little receptacle that I keep these twigs and things in. And I put a layer of water in there, and then I throw it in a wax paper bag and just kind of, I don't even close it completely. I just kind of wrinkle the end of it to
Right.
create, you know what I mean? To create that humid environment. But yeah, you're right.
Yeah. Cool.
Hey, Brandon, when you were preparing your slide, were you trying to get material from a specific area of this, or are you just basically getting just wherever you could, or is there a fertile surface there that you're shooting for, or what?
Ooh, can you hear me? I got a note that I was muted. Um, the center of the disc is what I'm going for, but these things are so small that
Yeah.
grab the whole thing and just smash the hell out of it, and you know, you're gonna get the fertile surface.
Okay. All right. I was just wondering. Yeah, I'd like to try. I haven't looked at Dacromyces under the microscope yet, and I guess usually there's larger species than this, but I'm going to give that a try next time I see it.
Yeah. They're I mean, super unique in that they have the tuning fork basidia, meaning that basidium splits into a Y and one spore germinates off the end of each terminal, if that makes sense.
Yeah. Right.
Very
Okay.
cool.
Yeah,
Also,
very cool.
this was on a maple branch. Dacromyces mostly grow on conifers. This was 100% on a hardwood twig, which is another identifying feature for minor. Not only the fact that they're minor, tiny, but most Dacromyces like conifer. This one is a hardwood sapro.
Yeah. Just looking at it, I would just have thought it was an ascomycete.
Mm-hmm. Yep, I thought the
Right.
same thing.
Brandon, I hope to one day try microscopy when I get a microscope, but is there a more delicate way besides smashing your sample to be able to see this? Would you be able to use maybe like, um, I don't know, one of those X-Acto knives or something to try to slice into it, or is that not even worth it because it doesn't come out right?
Okay, when I say smash mount, I use a razor blade to just kind of get the smallest piece of material that I can. So
Out of the center or like from the edge?
in this case, I was going for the center because I thought it was an ascomycete, and I thought that's where the fertile surface was, because that's what you're searching for. But it's a jelly fungus, as it turns out, so it's a bit basidiomycete. I think the entire thing is the fertile surface in that situation. So I think, to answer your question, it just depends on the mushroom that you're trying to dissect, right? But
Nice, thanks.
razor blades, tweezers, getting the smallest bit of material, and then smashing the absolute living out of it. That's kind of the process there.
Great, thanks.
Well,
Does
your
no one
local
else?
Bianca, if you ever want to look at stuff in a microscope, shoot me a call.
Oh yeah, you're near Bryn.
Farmingdale.
Very good. Sounds great.
Nice. Does no one else have anything they want to share? Because
Keep going.
well, I shared 5 things. I sent 10 things. So
And that means you have 5 more to share.
Oh,
no, I want to let
damn.
other people go if there are other shares.
Go for it.
I just have a puffball. We could save it. It looks like a puffball.
All right. Well, I'll go through this one briefly because it's a new one for me. And it has the common name common black earth tongue, and I find earth tongues that are verifiably vulnerable. They're on the red list, that are much more common than this thing. So how the heck did it get the name? The top half of these were actually missing when I found them in situ. They were either knocked off by an animal or conditions, whatever it may be. But enough of the hymeneum was present that I could get some micro done. And I thought it was going to be Geoglossum sphagnophyllum. But the paraphyses for these are so much different. The spores, in terms of overall shape, as well as length and width and segmentations on the spores, they all fit for Sphagnophyllum. But if you can zoom in on the apex of these paraphyses, they're kind of constricted. They almost come to a point. Yep. For Geoglossum Sphagnophyllum, as well as glabrum, they're subglobose at the apex of these paraphyses. These are all either straight or somewhat constricted, which,
Hmm.
Interesting.
Yep. Yeah, look at the one that I'm highlighting that you're looking at right there by the 4. It's in no way subglobose, right? It actually tapers to a broad point, blunt point.
Yeah, definitely tapering. I saw a bunch of earth tongues with the top halves missing as well in Wisconsin. I should have taken one home, but it was kind of
Yep.
weird.
Is something eating them or something, you think?
I think it's
Just
more something
kicked.
doctored over. Yeah, I think so. Only because I've never seen insects on any earth tongue. I've never, there are never larval trails. There's never like a slug munching on one, uh, anything like that. But can't rule anything out, I guess, right?
It would just be funny to, I don't know, if something's eating it, you know.
Maybe a deer or something.
Hmm. Cool.
My weekly earth tongue. All right, what do we all know about Bogbodia uda? So this was growing uh, under a footbridge, like a man-made. People, uh, screwed together a bunch of branches to make a little footbridge over sphagnum and peat bog. Um, and presumably this was growing out of some sort of moss, if not sphagnum. Um, you can see the edges of the gills are yellowish, where the actual gills are, dark gray, black. But that color change right on the edge is a Bogbodia uda thing.
I like that. It's like, whatever. What is that? 12, 15 that are maybe all the way up. I mean, there's really not a whole lot of gill up in there.
Mm-hmm. Yeah, a lot of short gills. Little velar material on the margin of the cap.
Hmm.
How many other members are in this genus?
0.
Oh, wow.
Yeah, this is what is that? Igor, what's the word? Mono something. It's the only
Monotypic.
monotypic. There we go. It's the only member of Bogbodia.
Very
It's a
cool.
monotypic genus.
And then, the edge of the gills kind of tell the story, give it away for me. I think I know this thing kind of. But I'm doing a study, so gotta actually do the micro, and the spores confirm this is Bogbodia uda. Um, Not only the shape, they're varicose as well, and then the size. They're pretty large. If you can zoom in on this one, this is a good example. Yeah, you can see they're kind of warty, varicose. It does have a germ pore at the apex up at the top there. I think you can make it out. And it's, narrowly ellipsoid, but with some angles, so it makes it amygdaloid. Amygdaloid? Is that the right word?
Oh, I don't know that one.
Amygdaline.
Almond shaped, right?
Yeah, amygdaline. Yes, almond shaped. Yes.
But Igor, do you know this one? You agree here? It's. I think it's more common than is reported. I know it ends up at our ID tables at 4As pretty often.
Yeah, I mean, I've heard this name and I've probably seen this at forays, but this is not the kind of mushrooms that are gonna grab my attention right away, and it's just an LBM. I'm not gonna, you know, uh, contradict your ID here because you're much better at these things than I am, or that I will ever be. Um, but the
Mm,
thing you said about monotypic, this is the only extant species from that genus, but it doesn't mean that there were probably more species, but they were just extinct.
yeah, or uh, misunderstood. Haven't Bogbodia as a genus probably hasn't been studied all that well.
Have you tried to look for any recent publications of on this genus? Any reports?
Everything. No, well, there aren't any papers that I am unable to unlock. Which those are the papers that I tend to focus on nowadays. It's like, oh, I can't unlock this thing. It must be the most important paper. um, but yeah, everything that I've read, it's monotypic, pretty easy to identify if those spores are over uh, 15 microns. All these other kind of bog associated hypholoma, uh, Gallerina, psilocybe type deals um, have spores that are much smaller. So I think that's
Okay,
where I think that's where the line is drawn.
right. So what are the? I mean, obviously, it sits in a clade of genera that are part of some family or subfamily or whatever. So what are the closest generic relatives of Bogbodia?
That I don't know. That's not my thing. Uh, Reading trees, at least, is not my thing. Um, but morphologically, the things that I just mentioned, then I suspect they're especially psilocybe because the spores are super similar to psilocybe spores. Um, but Hypholoma, Gallerina, Foliota, that's that's where these things are compared based on the description of Bogbodia uda. But
Okay, I think you're right in the money. I just opened up a page in Wikipedia and it says phylogenetically, Bogbodia is distinct from Hypholoma foliota and Leratomices, but it is part of the Strophareaceae family. So I
uh,
think all these things are Strophareaceae, in my
mhm,
understanding.
cool.
So yeah, you're absolutely right.
So I
Good
don't know
job.
if it's related, but the my little Latin Latin book, Udus, wet, moist, paludis. I don't know. Maybe it comes from that, since it's a bog thing.
Yeah, the name is really interesting. Actually, when I googled uh, I literally Googled, where does the name Bogbodia come from? And the first thing that came up was a Facebook entry from Maricel Patino.
Of course.
Yeah, who was also talking about how cool she thought the name was. So
So did you get any traction on that?
nah, I moved on.
Hmm.
I don't have Facebook, so didn't read the discussion.
So it says here the etymology of Bogbodia. It alludes to the tan color and occurrence in northern peat bogs, as do bog bodies.
Right. Yeah.
Huh.
And bog bodies are cadavers, right? Found in peat bogs.
Yep.
I know nothing about
To
that.
well, to preserve them, right? That was the practice at one time.
To be mummified, I guess, right? Yeah,
Yeah.
yeah.
Hmm.
Oh, I was muted. Sorry. Uh, Calling this one abrupta. Feel pretty good about it. Um, Pyramidal warts, big abrupt bulb, big old veil. The interesting thing about this one is that it was absolutely, if you can zoom in on the bulb, actually, yeah, it was growing out of a rotten pitch pine, which I'm not saying it's a saprobe, although it very well could be. Um, I just thought that was interesting, but the abrupt apex of the bulb and the pyramidal warts. I think I have a reference in my head for this particular species, Amanita abrupta, and I think that's this. What do you think, Igor?
I think you're absolutely right. Do you have a photograph of the well? How should I put this? The under the partial veil, under the
Hmm.
under the skirt, so to speak. You should see
Uh,
some fibrils that are attached to both the ring and the there you go, and the stipe, like
yeah,
struts. I call them struts.
yeah, yeah. I did see that actually.
This one hasn't probably expanded, but we often see those things. Like it's almost like when the ring expands, it just drags some of the stipe tissue with it or something.
Interesting. Yeah,
But yeah, you see, there's something going on, on the inner surface of the ring.
I absolutely took note of that. There was some. I wrote it down in my book as some action under the uh, under the veil.
Under the veil, right? Yeah, under the annulus. Yeah, I think this is. I mean, this is one of the easiest Lepidellus to ID, along with polyperamis and maybe to some extent subcoccori in the New Jersey Pine Barrens and
Mm,
elsewhere. I mean, it's pretty distinct. I mean, what else? What other species of Lepidella have this kind of a bulb morphology, which is very
right.
abrupt, uh, kind of rotund, and you see how you get this broad shoulder is just completely flat.
Yeah.
That's
Yeah.
like a prototypical of the species.
Cool. All right. Nice. I think that might be the one Amanita that I might be able to figure out every time. I think I know that one, which
You
is
know,
famous last words. But
no, I honestly the past 2 weekends I have had moments with two different people where they knew a thing and it makes me laugh every time. because there's a difference between you can figure something out or I think I know, and I am so like I so know it. Uh, at Swartzwood, I had uh, a resinous polypore in my hand, and Lila walks past me, and in mid-stride, she just turns and looks at it and goes, "Ooh, resinous polypore. Those are good." And then takes a half a step back and goes, "Let me see the underside of that thing." Yeah, 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 resinous polypore. That was good. And then I showed her a picture of the log of all the other ones next to it, and she's like, "Oh yeah, definitely." Because it clearly was, she so knew it, like she was walking past and knew it. And then this past weekend, I had one, a dull orange mushroom, and a couple of people had found uh, jack-o'-lantern. And I'm like, nah, this is different. Nina comes walking by and same thing, like, ooh, that's a nice fall chanterelle. It's just it's awesome when people they have it hands down, like they just know it.
Yeah, well, I mean, it's the people that you just named. It's not crazy to hear those names. They know things. You know what I mean? Yeah, I ask Igor a thing. I know he knows it.
But yeah, but it's funny because like it's it's the difference and you can you know and like
The
without
different,
even a thought,
the,
like you know
yeah,
they know.
the difference between like, I don't know what this is, and then, oh, yeah, from a mile away, I can ID that thing, right?
Yeah, my list of things like that are super short. But even for me, like I can look at like this tiny handful of things and I know exactly what it is.
Mm-hmm. Yeah, man, it's knowledge base, pretty, you know.
Hey, Brandon, I
Yeah,
had a quick question about the last one. Is that Roanokenses also?
yeah, right. Which one did we just look at?
Abrupta.
Abrupta. No, that's
Padella? Is that what section is
yeah,
that?
yeah,
Well,
no,
well,
that's Lepidella. That's Lepidella?
well, it was broadly placed into Lepidella sensibus, but now Lepidella only encompasses just a handful of species that are not mycorrhizal. So it's also subgenus Lepidella, section Lepidella. So all these amanitas that are saprobic, of which there are very few in the world, maybe, I don't know, less than 2 dozen, fall under section Lepidella. Everything else that's used to be in section Lepidella, most of it moved out to section Roanokenses in subgenus Amanitina.
Okay, that's helpful. I thought Lepidella was just gone completely, but
Well,
it's
it's sensu boss, right?
Sansubas. So that's like this is before that seminal paper by the Chinese from 2018 that took a much closer look at the whole genus worldwide, and they used several markers, genetic markers, to build their trees, including several protein coding genes. So this is where they changed the taxonomy of Amanita, wherein there was a necessity to split up section of Lepidella because it was polyphyletic, in the sense that it didn't, there were things that were scattered, about the tree of Amanita. They were not from a single source, a single point of origin in the tree.
So,
So
so to that more poignant question, would that one? Wow, no, it wouldn't be Roanokensis, right?
no, it would be Roanokensis. The other choice
Oh, it
is
would.
Strobylliformes,
Strobylliformes, that's the
yeah,
other one. Okay.
which we don't know what the extent of the content is, species list in that section, because so few have been sequenced. But for instance, Rhopalopus is in that section, and probably Ravenellii, and probably Rhopalopus too. So to me, Ravenellii looks more like a mainstream kind of Roanokenses type of Amanita, but apparently it's in a different section. So all these Lepidellas got split up between 3 different sections now, located in 2 different subgenera within Amanita.
Gotcha.
But you just have to
Cool.
remember that the original Lepidella only circumscribes a handful of saprobic, non-mycorrhizal Amanitas at this time.
Okay. Thank you.
The bulk of it is going to be in a section Roanokenses.
Cool.
Maybe 70%, something like that.
And then we have I'm calling it crenulata. This cap is quite kind of peachy, not quite yellow.
There you go.
But that right there, right? Yeah, crenulata.
Right there. We're
Yeah.
looking at it right now.
Yeah. Okay. Cool.
That deposit on the upper part of the bulb, the champagne.
Yeah, the yellow,
ring?
the pale yellow band, right?
Of velar remnants, correct. This is universal veil, correct.
But the cap color I thought was interesting. I mean, it's kind of peach colored. But that look at the color of that UV material. Yeah.
Yeah, it probably just was affected by the weather. It just was, I think it's drying out. And but to me, the color of crenulata, the cap of it, always, I mean, let me put it this way: when you look at it too much, you would see sort of pinkish tones, sort of in that tan color showing up sometimes.
All right. Cool.
I mean, does your camera, alter the colors sometimes? Do you observe?
Sometimes, but not in this situation. Actually,
Yeah,
I found this. I found this at like 6 o'clock in the Pine Barrens, so it was like golden hour. It was perfect for photographs.
Right, and remember that when you take photographs toward the end of the day, they tend to be more red shifted. They
Yeah.
tend to be warmer, redder colors overall. But
Okay.
you're saying that your eyes saw the same thing that the picture shows, that the color is
Yes.
true.
So maybe the color is not true now that I'm looking at it. I definitely thought it was more salmon colored, and this does look a little more red tinged than salmon. I just noted that it was not a pale yellow, right?
Well, I would just call it tan. I don't think they're yellow. They're darker yellow brown. Not sure. I'm terrible with colors. So you have to use like a color chart. And speaking of colors, there's a standard reference that mycologists use. I think it's a book by Methuen. It's called Methuen um, uh, Catalog of Colors or something like that.
Oh, okay.
And I suggest you purchase that book. I mean, I think it's out of print, but you can buy used copies in good condition. And let me actually tell you what the name of the book is. Yes, it's Methuen Handbook of Color. It's a British publication, and the authors are Cornerup and Wanschler. I can send you more information. So when you read papers and people describe colors, they give like codes, like something like 8A4. It comes from that book.
Nice.
I wonder if that's the same as the because there's like a universal chart that like industry uses for like colorants of all kinds.
Well, there are several of them, and I actually own a chart that has all these AOC numbers, and I think AOC stands for online cover chart or something like that. Where did I put this thing? I can find it, but anyway. Oh, well, here it is. Yes, it's the online auction cover chart. So that would be also a good reference, because I think it has a lot of interesting colors there too. So yeah, but the papers use the Methuian Handbook of Colors mostly.
And I will come back to something that Igor, I remember you telling me when I first joined the club, and has been echoed from every other person that I've learned anything from about fungi. Color is an unreliable resource for fungi. You know what I mean?
It is.
A yellow thing can be kind of pinkish or a reddish thing can be kind of yellowish depending on conditions. So that always stuck with me. Color is one of the least confident things to hang your hat on when identifying fungi.
Everyone sees color different.
Mm-hmm.
Not to mention that there are 1,000 shades of one color, and
You call a thing red,
so
it's
how would you call this? I think this is some kind of a yellowish brown color. It's not yellow nor is brown.
all right. Well, let's just call it ochre. How about that?
Yeah, but even ochre, if you look at the definition of ochre, there's a whole spectrum of ochre shades.
Yeah. So I would describe this as yellowish brown, personally, or brownish yellow.
Yes.
You know, but with obvious hints of red, right? There you go, all over the thing. Enrolled cap, tight gills. This is how it was found in situ. This isn't necessarily staining, although it probably is staining from outside factors, but that staining was present when I found the thing.
Did something bleed on it?
Say it again.
Did something bleed on it?
It's the thing that is bleeding, or at least presenting red. If we go to the next photos, you can kind of see it's present in all aspects of the mushroom. Yeah, this is the in situ photo. Super, inconspicuous, kind of buried. And then, yeah, there you go. It's marginate. If you can zoom in, you can kind of see these spots on the gill edges, the gill faces, that all ultimately go to red. So I think I know this is Russula ventricoccipes, but maybe I'm wrong, and there are other options out there. I did find some red, then black staining Russula specimens this day. This thing never went to a black stage. Didn't even really stain red. It just kind of kept this coloration the whole time. So, anyone have a alternate ID or a supportive comment on ventricoccipes?
I'm
I
not
concur.
used to Russellas having the separation from the stipe with the
So
No other Russula looks like this one.
Well, that's why I'm saying I'm not sure. I mean, I'm not a Russula expert or even close, but I thought all Russulas had attached gills to the stipe.
Yeah, they're attached, except that this thing is just sort of expanding and ripping itself apart.
Yeah, over here.
Okay. Yeah, okay, I see it.
And
Yeah, I
then
mean, it's one of a kind Russula. You would not find this outside of the outer coastal plain, Atlantic coastal plain. So you would only find this in the Pine Barrens, and it's fairly common. And it's a very unique looking Russula. So once you see it a couple of times, you will never forget the name. This is one of the easiest Russulas to ID, period. Anywhere.
yeah,
And
yeah,
as far as I
and
know, they smell bad and they're not quite edible at all. They're probably even poisonous. I wouldn't be surprised if they're poisonous.
one of the fetid Russula, right?
I have to look up which section or subsection it's in. Possibly.
But yeah, I wanted to share that. I'm not sure. Have we seen that here before? Maybe I shared it a couple years ago. But yeah, you're right. You were Pine Barrens thing. You only see it there. So if you're not hanging out in the Pine Barrens, where it is common, uh, it's uncommon outside of the Pine Barrens in New Jersey.
yeah, I don't think I've ever seen anything like it. It's cool.
Yeah, Kuko says here on the page for on his page for Ventricocypes that it is a member of the Theta Drusilla group. And here's the link. You can look it up.
Cool. I feel like I've been sharing for days. Uh, anyone anyone
I'm
else want to
curious, actually, did you split that one apart?
go? Yes.
Any pictures of the? I'm
No.
assuming it might have been eaten on the inside, because it looks older.
Well, no, it was it had a bit of a chambered thing going on, which many Russula do. But yeah, there were little pockets that were eaten out.
So this was like
Thanks, Brandon.
a softball, and I just noticed they're all over the neighborhood right now.
Calvatia?
I don't know. They're all about this same size. They all seem to be growing in a spot where it's inappropriately sunny all day long.
Yeah,
And some, they're just it's just random on a couple different lawns around the neighborhood.
Did you cut any of those? Did you slice them up?
This is the best looking one, which I did not touch, but there was another one a couple feet away from it that I mushed open a little bit with my hand, and it was like had like a green tinge, but it clearly hasn't started going all the way yet.
And the size of this was like size of a fist?
A little bigger, like a full softball.
Okay.
well, probably like Cyathoformis.
Yep, that's what I think it is.
Yeah, Calvaceous, Hathiformis, or yeah.
Yeah, these things kind of confuse me. If you brought it up and I could see that granular, right? No, skull-shaped thing, right? Isn't that the cyaniformis?
That's a
No, that's
craniiform.
craniformis.
Oh, yeah. Okay. Yep. My bad.
I think they have more of a kind of a stalk appearance. You know, they have an elongated base.
The one that I pulled away some grass on a different lawn, it had a bit of that. And I don't know if they're all like exactly the same, but they all came up at like the same time.
I think once it goes bad, is it craniformis has the purple spores, and then otherwise they're very very difficult to tell apart.
I know I want to like I keep revisiting them and I'm like, oh, they're still there. Trying to let it go all the way, but I just know how people are, and they're right there, like ripe for the kicking.
Okay,
How big were they?
They're all real similar to like softball to a hair larger than softball.
Yeah, that's pretty big.
so Cyathoformis would have the purple spores, and then craniformis would have yellow, yellow brown spores, and has the narrow sterile base.
Yeah, I'm trying to hold out and like not touch anymore,
They both
until
might
they
be
until
out.
they go further so I could see what color spores are. But if the lawn guy shows up, forget it.
Mhm.
Yeah, that's hilarious.
I'm so aggravated by it.
I ran out of my apartment when the landscaping crew showed up recently. I've been watching this thing. I'm going to collect it before you get to it.
I know. On my way home from work, I took like the extra detour and went around the block. I'm like, all right, they're still there. Like I'm creeping on this thing.
Yeah, these are fun though, because you can see them from the car. It's great, you know.
Yeah. Yeah. Well, that's all I had. And I'm gonna keep driving by that thing.
Well, collect it at some point and get us a section, man.
Yeah, well, that's the thing. Like if I collect it too soon, isn't it like not worth it?
No, because you can figure out if it has that sterile base or not, right? You know, what is it? Craniformis, the skull one
Yeah.
has that shape. Um, Cyathiformis, what is it? Cyatha
So
something?
so you're
Yeah.
saying at the base of this thing, there's a detail?
Yes. You said you had seen
It
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 though
had
on
like
one of
a
them, right? A
it
stem
skinnies
has a
up like almost like a little stipe-ish. I don't know what else to call it.
Oh, well, they'll all have like a little root connection to the earth. Like all those giant puffballs will have a little, I mean, it's like a little rooting down part that connects it to the earth, like plugs it into the planet. But the cranioformis will have it looks, kind of like uh the was it the limbic system of the brain, so it caught like, or your neck, like a neck on the head.
Hmm.
Yeah,
So
like an obvious sterile base, as opposed to just kind of mycelium connecting it. I like how you described that, connecting it to the earth.
yeah,
Plugged in.
plugging it into the plant. Yeah.
Yeah, yeah,
Mhm.
plugged in.
Yeah.
Love that.
Yeah.
Yeah, so okay, so all right. I'll drive by tomorrow after work, and if it's still good, maybe one of them that I messed with already. Maybe I'll grab one of those and tip it over.
Well, if there's a bunch of them, collect
Yeah,
one and see what happens later.
yeah, yeah. The one of the ones I already messed with.
Yeah, I think it looks like the Cyathiformis from here, but we'll see. Yeah. Those are fun to find. But you said it was kind of yellow or green already inside the one you.
The one, yeah, and like a warm pizza dough. Like, man, that thing was squishy.
Mm-hmm. Yeah. Hmm. Sounds like it was going bad. I mean, yeah. So maybe those yeah. That'll be interesting. Yeah.
Every
Yeah,
year,
I keep holding out to like maybe it'll be there long enough to get into some spores.